Hi, so I'm Daniel Dempsey. I'm an assistant professor in the Department of Pharmacology, Physiology, Biophysics, and Dermatology. And with that, my whole 4 minutes is done, but I just kidding. So we're a chemical biology group, so this will be a little bit different. I promise to keep the chemistry to a minimum. But one of the unique attributes of our group is we're able to actually engineer proteins that have unique chemical functionality. Usually we do this to study post-translational modifications by installing site-specific and stoichiometric modifications. But we also frequently make chemical probes, full-length protein probes, to be able to identify interesting interactors or enzymes that may process these proteins. And we're doing this in 2 general areas. The first is what I'm going to talk more about today, which is unlocking the secrets of the ubiquitin-proteasome system and its role in age-related diseases. And the other is we're studying how post-translational modifications regulate important proteins in RNA biology. So I'll just talk about the first project today. And so here I'm just showing, so one of our research goals is to identify vulnerabilities within the ubiquitin-proteasome system to target with novel therapeutics. So here I'm just showing a basic schematic of the ubiquitin-proteasome system, very simple illustration. And so proteins get ubiquitinated by a class of enzymes called ubiquitin ligases. And they install this small protein or polypeptide that's 76 amino acids long onto proteins, and this serves as a molecular stamp to have the protein shipped to the proteasome to be destroyed. So that's one way that the cell uses to clear out proteins. But there's a special class of enzymes called deubiquinases, or a special class of proteases that will remove ubiquitin from proteins. And so if ubiquitin ligases is serving to destabilize proteins, deubiquinases could be serving a role in stabilizing proteins. And we think that this class of enzymes could be a unique class of enzymes to target with novel therapeutics to try to destabilize proteins that drive disease. And so the question is, how can we leverage the system to destabilize proteins that drive age-related diseases? So as I said before, we're a chemical biology group. We like to use chemistry to identify things that directly we can target. So how can we identify deubiquinases that directly stabilize proteins? And so I'm going to show you one of the probes we're designing now. We've done this with a number of different proteins successfully so far that are vital for human health and disease. But this is the general makeup of the probe that we have made. Is we're making a full-length protein where we put ubiquitin site-specifically and stoichiometrically at its relevant site that we know is important for its stability. But instead of having the natural ubiquitin linkage, we're installing this chemical warhead instead that will covalently trap the ubiquinases that target that protein, and then we can identify them by mass spectrometry. And we've done this successfully for a number of different proteins. So where are we going with this now? We're advancing innovative methods, chemical approaches to identify and characterize deubiquinases that target proteins that cause disease. We plan to evaluate their contribution to the pathogenesis of disease. We're going to evaluate other substrates those deubiquinases target, and then we're going to start to design and screen for inhibitors to be able to target these deubiquinases disease. Thank you.